hold on for a second. Sir, can you please click on the hide button so that it will be.
Oh yes, in the bottom of this, yes, it is gone. Okay, the technical team, we are ready for the streaming. Yes. Okay, a very good morning, everybody. I welcome you back again, and we are going to start the second lecture of the day. Our second speaker is Dr. Prasanta Saini. We warmly welcome Dr. Prasanta Saini in this scientific session today. Dr. Saini is serving as the officer in charge of the ICMR Vector Control Research Center field station at Kottayam, Kerala since 2019. After completing his PhD from Vishwabharati University, he started his postdoctoral work in Entamoeba histolytica at the ICMR National Institute of Cholera and Enteric Disease, Kolkata. Later, he shifted to Bose Institute, where he worked in leishmaniasis immunology. Dr. Saini joined the ICMR Vector Control Research Center, Kottayam, as Scientist B and presently working as an adjunct scientist at the Department of Molecular Entomology. He received various prestigious national fellowships, including ICMR Centenary Postdoctoral Fellowship and UGC BSR Meritorious Fellowship. He is the member of Indian Society for the Parasitology, Indian Science Congress Association, and the Society for the Vector Ecology. His research interest includes, but not limited to, morphological and molecular characterization of sandflies in India, their immunology and signaling mechanism, and epidemiological studies in leishmaniasis in Kerala. He has 18 international and 2 national publications. We cordially welcome Dr. Saini for his talk today on atypical manifestations of Leishmania donovani in the Indian subcontinent. So it's over to Dr. Saini. You please continue. Oh, I have a announcement to do. So there would be a reminder 3 minutes prior to the end of your talk. So Dr. Saini, please continue.
Can I start my presentation?
Yes, you can start.
So very good morning to all of you. Thank you, Doctor, for my brief introduction. And I want to convey my sincere thanks to the organizer for inviting me in this webinar. And I get an opportunity to present our work in this web platform. Now, the title of my presentation is Atypical Manifestations of Leishmania donovani in the Indian subcontinent. Here, I shown 2 patient picture of leishmaniasis. This is the visceral leishmaniasis patient picture, and this is the cutaneous leishmaniasis patient picture. And both these 2 disease manifestation caused by Leishmania donovani. In general, the endemic zones of India and its subcontinent, the visceral leishmaniasis is caused due to the Leishmania donovani. But in some pockets in India and its subcontinent, the same parasite causing the cutaneous leishmaniasis. For that, we are continuing our research on that topic, and I am now going to present how we identified that Leishmania donovani causing cutaneous leishmaniasis means it causing atypical manifestations of leishmaniasis. I am not going to describe in details about the introduction of leishmaniasis. Very briefly, I am describing the introduction. First of all, that leishmaniasis is a very old disease. Means this disease started from long back, around 1st century AD. And after that, in different tropical area are infected by this disease, and in local different region, this disease named differently, just like Kalazar, black fever, Kaladu, Aleppo boil, Damdam fever, and in Kerala it is called as Karimpeni. Now that these parasite name given by Ronald Roche by the honor of the two pioneer famous scientists William Leishman and Charles Donovan. Now, in India and entire globe, around 30 species of Leishmania parasite infecting the human and in different mammals also. And out of that, 21 are pathogenic for causing the disease. And in Leishmania donovani, it works as a complex. In that complex includes donovani, infantum, and chagazi, and Mexicana includes three species, that is Mexicana, amazonensis, and venezuelensis. And other major parasites are Leishmania tropica and Leishmania major. And in this disease, sandfly play as a vector. And these parasite means Leishmania donovani, it have major two forms. That is, one is promastigote form, which is present in sandfly, and another is amastigote form, which is present in human and reservoir host and other mammals. Now, in sandfly, throughout the globe, around 500 species are reported as the different area that sandfly are prevalent in different area throughout the globe. Out of that, 30 species play as a vector in different parts of the world. And in India, Phlebotomus argentifus and Phlebotomus pepatasi are the major vector for Leishmaniasis. And these sandfly are passing their life cycle and complete their life cycle through different life cycle stage, just like egg, larvae, pupae, and it's ultimately it become an adult. Now, I want to tell the different infective stage. Means in human body, the infective stage is the amastigote form, which is an intracellular parasitic form, and it that amastigotes multiply in infected cell and infect different tissues. Then, when an sandfly taken the blood meal from the infected host, after that, in the gut of the sandfly, that amastigote turns into promastigotes, and again that promastigotes keep ready themselves to infect another healthy host. These are the general life cycle. I am not going in details about the life cycle of the Leishmania parasite. And now I want to tell the epidemiological situation throughout the globe about Leishmaniasis. Around 0.7 to 1 million new cases are reporting in every year. Out of that, 20 to 30,000 deaths occur annually. And these are the figure how that visceral Leishmaniasis are occurring throughout the globe. These are the figure for cutaneous Leishmaniasis, and that most susceptible hosts are that HIV co-infected patient. Now, this deep color area are mainly prevalent for visceral Leishmaniasis. Mainly in India are highly endemic for visceral Leishmaniasis, and other than that, Brazil, Ethiopia, Kenya, Somalia, and South Sudan and Sudan are also reporting these visceral Leishmaniasis cases. And these are the cutaneous Leishmaniasis endemic areas, and mainly this type means cutaneous form of Leishmaniasis mainly found in Afghanistan, Algeria, Brazil, Colombia, and Islamic Republic of Iran. There are several cases of cutaneous Leishmaniasis are reporting, and we also have some collaborator in that region, and in Pakistan, Peru, and Saudi Arabia and Syria are highly endemic for cutaneous Leishmaniasis. Now, that Leishmaniasis have three major forms, that namely visceral, cutaneous, and mucocutaneous Leishmaniasis. The visceral Leishmaniasis is mainly caused by Leishmania donovani, Leishmania infantum, and Leishmania chagazi. These are the typical manifestation caused by these three parasites, and that manifestation is visceral leishmaniasis. And Leishmania tropica amazonesis is major. Typically, it causes the cutaneous leishmaniasis. But we are working in atypical form of leishmaniasis. So why we are calling this is an atypical? Because Leishmania donovani, it's causing the cutaneous leishmaniasis in the Western Ghat area of Kerala and few pockets of Himachal Pradesh. In that area in India, reporting that Leishmania donovani causing cutaneous leishmaniasis. And along with cutaneous leishmaniasis, few of the patients are also infected by this parasite and manifests as visceral leishmaniasis. So this is the major point that we are telling, that Leishmania donovani causing cutaneous leishmaniasis. Now, in globally, that atypical manifestation of leishmaniasis are denoting here that round structure. Means these Leishmania tropica causing VL, Amazonesis causing VL, major causing VL, infantum causing CL, Chagasi causing CL, and Leishmania donovani causing CL. These are the colored area and noted area where that atypical manifestation of leishmaniasis are prevailing. Now, in Indian scenario, that visceral leishmaniasis endemic zones are mainly Bihar and adjoining Jharkhand, West Bengal, Uttar Pradesh, and adjoining country like Bangladesh, Sri Lanka, and Nepal. There are several visceral leishmaniasis cases are reporting, and cutaneous leishmaniasis endemic zones are Rajasthan, and in Indian scenario that Kerala and Himachal Pradesh are mainly causing the atypical leishmaniasis, means Leishmania donovani causing CL, and in adjoining country that Sri Lanka, and in few cases also reported from the Nepal also reporting few of the atypical leishmaniasis. Now, recently, means last 2 years, from the Western Ghat belt of Kerala, we have identified around 24 visceral leishmaniasis cases and 41 number of cutaneous leishmaniasis cases from the foothill of Western Ghat belt of Kerala. Means in Kerala that elevation are in different. Means Kerala have different land structure, that is highland, footland, midland, and lowland area. And all the cases, whatever we are reporting here, and what are the manifestation we detected from Kerala, all the population are inhibiting in the foothill of the Western Ghat area. And from there, by using different genetic marker, we identified that the causative agent of the parasite is the Zymodium 137 of Leishmania donovani. Means this particular strain of the parasite, from 9 CL case and 4 VL case, we identified that this disease is caused by Zymodium 137 of Leishmania donovani. Now, how we have diagnosed those patients? Means based on symptoms, first we diagnose, preliminary diagnosis we perform for the VL patient by RK39 strip test and histopathological examination. After that, kDNA gene amplification. And in case of CL, we identified by histopathological examination and minicircle kDNA gene amplification. Because in case of CL, that RK39 strip test does not work. Now, after diagnosing those patients, we forwarded those patients for treatment, and they are getting treatment from the Command Medical Hospital. And after that, in our research aspect, we analyze the different marker to know what are the species involved in this infection. For that, we from the VL patient, we have collected the bone marrow sample as well as refined biopsy sample, and from the VL patient. And in case of CL patient, we have collected the skin aspirate from the area where the lesions are present for cutaneous leishmaniasis. Now, these are the cutaneous leishmaniasis infected patient. That lesion is like this. These are the lesion how a cutaneous leishmaniasis patient we are getting, and it that lesion structure, it's very difficult to identify that this is the cutaneous leishmaniasis. But our expertise research group and our we have some expertization that by we can suspect that these are the maybe the cutaneous leishmaniasis. After that, we are collecting the sample and we are doing our diagnosis in lab. Then these are the visceral leishmaniasis patient photo. Recently, we diagnosed these two patients from the foothill of this area, and we proceed for the parasite identification. For that, first of all, we from the sample of BL and CL, we have amplified the 3' UTR HSP70 gene, and after that we proceed for restriction digestion of that gene product, and it shows clear bending pattern, means defined bending pattern on 450, 170, 125 and 25 bp. But 25 bp band is very faint, it cannot visible here. So this bending pattern denoting that this infection is caused due to Leishmania donovani complex, means in this complex this infection may occur due to either Leishmania donovani or Leishmania infantum. Then we want to know what are the particular species for this infection. For that, we have amplified that HSP70 gene, and that gene product proceed for sequencing, and we found that in the position of 434 there are presence of cytosine, which denotes this infection is caused due to Leishmania donovani. But if it in that particular position thymine is present, so that indicate this infection is caused due to infantum. But we got only cytosine, so we can confirm that this infection is caused due to Leishmania donovani. And in different sample, we made an bioinformatic analysis, and it comes in a single clade of all the sequences. So we can confirm that this infection is caused due to the Leishmania donovani. Now we want to know what are the particular species which involved in this infection. For that, we amplified that 6 PGDA genes, means 6 phosphogluconate dehydrogenase gene amplification we have performed, and in that 976 position, if there are presence of adenine, so it denotes that this infection is caused due to MUN2. But in our experimental set, in that position, we got that guanine. So it's confirmed that this infection is caused due to the MUN37 strain of Leishmania donovani. And all the identified species, whatever we collected samples and proceed for identification, those comes in a single group, and from Sri Lanka and China group and Bangladesh group, whatever they also done such type of experiment, it comes in a single clade. So we conclude that this MUN37 strain of Leishmania donovani causing the cutaneous leishmaniasis in the Western Ghat belt of Kerala. Now, these infection, which is the vector? To identify the vector, we collected the sandfly sample in and around the patient house, around 0.5 kilometer radius. We collected the sandfly species and we dissected the from the female. We dissected the gut of the sandfly and we amplified HSP70 gene for identifying that parasite species, and we got that amplification for that particular gene, and it comes in a single clade whatever we got in patient. So we can tell that sandfly is the vector for this disease. Now we want to know what are the particular species which causing this means which play as a vector for this disease. For that we done the DNA barcoding analysis from the same isolated DNA sample by amplification of CO1 gene, and that CO1 gene barcoding analysis shows that this vector is Phlebotomus argentifers, and that how many sample we got infection all comes as a Phlebotomus argentifers. Now we want to know that is there any animal involved in this infection, means can any animal or any species play as a reservoir. For that, in that patient house and in and around whatever the animal present, means cat, dog, rodents, from there we have collected the blood sample, and from the blood sample we again amplified that HSP70 gene, and we got that amplification and that sequencing analysis. When we have done, then we saw that HSP70 gene sequence from that reservoir host, means from that dog. From dog, whatever we got that amplification, it comes in a single clade, means that sample amplified from human, infection got from sandfly, and infection got from the blood of dog. Each and every gene sequence of HSP70, it comes in a single clade. So it confirms that HSP70 gene amplification, that all the involvement, means human is the host for this disease, sandfly are the vector, and Phlebotomus argentifers is the particular species of vector, and the dog play as a reservoir for this disease. And this is the manifestation here, that visceral and cutaneous leishmaniasis both caused by that Leishmania donovani MON37 strain causing this disease, and dog play as a reservoir, and Phlebotomus argentifers is the vector for this disease. These type of experiment recently we published in Emerging Infectious Disease, and such findings are very important in present National Leishmaniasis Elimination Program. Now I want to tell another story about our research in vector biology. So in that vector biology research, we have performed prevalence of sandfly in different endemic and non-endemic zones, means different biogeographical zones of India. We are collecting samples and we are identifying that what are the species, means by genome monitoring method we are identifying that infection also, and what are the determinants, why only the sandfly playing as means in sandfly Phlebotomus argentifers only play as a vector for this disease. And some findings we got recently that I cannot describe here because it is an unpublished data. We incriminated one new species from the Western Ghats wave, what play as a vector. I don't know it actually cause the atypical manifestation or not. As we didn't publish, I cannot show all those data, but in preliminary information, I can describe here that some determinants play as a major role for this disease. Means these are the blood taken by that infected means sandfly from infected host, and after that it mature inside the gut, and different life cycle stages are there. Just like from infected host, it takes the blood from infected host as amastigote, then procyclic chromastigote, ectomonad, leptomonad, heptomonad, metacyclic chromastigote are infecting the and healthy host. In between, several life cycle changes are happening inside the gut of the vector that we are doing in our lab. We are finding several new methods and new molecules also we have means immune molecule also we have identified from these evidences. And why only the sandfly play as a vector means argentipus, that some evidence also we found in our research work. And finally, some natural barriers, how they survive in only in the sandfly. For that, some natural barriers are there, like saliva, blood meal, digestive enzyme, then abdominal midgut and stomodeal valve, all are involved to facilitate the establishment of infection. For that, there are another major evidences are there that gut microbiome also a major determinant for establishment of infection. Now, here I shown some model. This is the conducive environment of tropical climate. This is the pathogen and this is the susceptible host, where all three are matching. That is the disease transmission. Now these are the I want to acknowledge the funding agency like ICMR, ICMR-VCRC, Indian Medical Association, and our strong collaborator is Thrissur Government Medical College Trivandrum, and Department of Health Service, Government of Kerala. These are our research groups. Without them, I cannot execute such type of work. And our major benefit of research, we are collecting sample from the field and we are doing research in lab. In between, nothing means we are doing our research on field collected sample, so it can easily get benefit the society. That is our major benefit in this research. And I am very much grateful to our institutional different colleagues who help us lots to execute such type of an experiment and research. And thank you for your patience. Thank you so much.
Thank you, Dr. Saini, for your excellent talk. May I request the moderator to take over to continue the question and answer session?
Thank you, Dr. Mandal. Thank you very much, Dr. Prasanta Saini, for this wonderful presentation. Now it's time to take some questions. So
one minute, just a minute. Dr. Junaid Gibran Javed is my friend, and he worked with me, and we worked together around 3 years. Around 3 years, we worked together.
Yeah, more than 3 years.
Division of Molecular Medicine under Professor Subrathan.
Thank you very much, Prasanta Saini. And now it's time to take questions. So I will request the faculty member of DLS and the member who have joined Google Meet, if they have questions, then they come up with that. Any questions?
Yes, I have a small question. Dr. Saini, it's a great talk. I just have a small, I'm just curious, these atypical leishmaniasis, which is caused by Leishmania donovani, do they show any splenomegaly and all those typical manifestation of visceral leishmaniasis?
Yes, all type of manifestation, just like visceral leishmaniasis, they are in manifestation base, there is no difference. Only the involvement of pathogen are different. Means in atypical manifestation, mainly here I'm described here that Leishmania donovani caused the cutaneous leishmaniasis. That manifestation is little bit different. Means Leishmania tropica mainly causing the cutaneous leishmaniasis in India and subcontinent. But when it caused due to donovani, that lesion wise in cutaneous leishmaniasis little variation are there. But when it causes the visceral leishmaniasis, symptomatic based no different we found in the patients.
Right. So no, my question was, yeah, and that's the lesion wise. My question is, when they are doing the cutaneous, like when the lesions are showing up, do they also there the spleen is affected, or like
No,
how
no,
it
no,
happens?
no, 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 no.
Oh,
And
that.
I have some evidence that one patient we identified, that patient when they infected by visceral leishmaniasis, for during the study of case history, when we collected the case history, that patient informed us that before 3 years he have some lesions in the exposed part of the body. Means that time he didn't bother about that lesion, but we
Mm-hmm.
suspect that that time that lesion may be caused due to that parasite, and after some time again that parasite didn't completely remove from their that that patient body,
Body.
and after
Uh-huh.
some time it infect that visceral organ. This is the situation.
Yeah, that's very interesting, actually. Okay, that. Thank you so much.
Thank you.
Thank you, Dr. Mukherjee. Is there any question from member joined through Google Meet? If not, then I'm moving to take questions from YouTube Live. So there is first question asked by Amlan Banerjee. What conditions should be considered in the different diagnosis of mucocutaneous leishmaniasis? Dr. Prasanta Saini.
That in case of visceral leishmaniasis, easy and strip test diagnosis is RK39 strip test. But gold standard method is histopathological examination, means to find out the LD bodies, and after that we are amplifying the kDNA gene of from that patient sample, means from the bone marrow and trephine biopsy sample. These three methods generally we are following for preliminary diagnosis. When all three we are getting positive, immediately we are referring those patients for treatment.
Thank you. Another question asked by Orithro Bhattacharjee. How can individual physiological conditions influence
Please.
susceptibility to leishmaniasis?
Sorry.
How can individual physiological condition influence susceptibility to leishmaniasis?
That Can you repeat your question, Dr.
Yeah,
Junaid?
sure. How can individual physiological conditions influence susceptibility to leishmaniasis?
That susceptibility means in a environment when all factors are matured and all are together, then only got a particular environment to infect the patient. That such type of clear evidence we didn't establish for that.
Okay. Another question is asked by Shubhashish Indo. Why atypical leishmaniasis is observed in some particular part of the globe?
That is a very great question. Means here, mainly in Indian subcontinent, two pockets are there where the cutaneous leishmaniasis is, means atypical cutaneous leishmaniasis is occurring. Here in the foothill, in between the highland and midland, there are foothill area. In that foothill area, among the tribal population, most of the cases we are getting here. Then recently, whatever we published in that published paper, we also mentioned that when a patient case history we are taking and noting that time these cases are indigenous cases, means they never exposed to any population from endemic zones, but those patients are going inside the forest and they are they stayed another 15 to 1 month to collect their livelihood material from the forest. We don't know inside the forest some parasite circulation are there or not. And these atypical manifestation also found in the Himachal Pradesh belt, means they also recently published in the same journal that 137 strain of Leishmania donovani, it's causing that atypical cutaneous leishmaniasis. But the mechanism till now not established. We are working on that. Recently, we have a project funded by ICMR to find out the mechanism about this disease manifestation.
Thank you, Dr. Shani. Could you please stop presenting and so that the participant can see you? The next question is asked by Avik Mukherjee. Is there any specific molecular marker either in host or in the parasite to differentiate between classical and atypical leishmaniasis? Hello. I think Dr. Saini somehow is got disconnected. So we are requesting him to
Please.
join again. Yeah. Yes, Dr. Saini. The question is asked by Avik Mukherjee. Is there any specific molecular marker, either in host or in parasite, to differentiate between classical and atypical leishmaniasis? Dr. Saini, I think you have to unmute yourself. Dr. Saini, please unmute. I think the voice is still not clear. Yes, Dr. Saini, please unmute yourself. I think there is a problem with some technical problem is going on that you are not able to unmute yourself. You can check from your side first. Meanwhile, all the present participants, here is an important announcement for you all that we have a feedback link that is present in every our every lectures description box, and all are same. So it will be activated after 12:30 p.m. today. So please fill up that feedback form so that you will get the e-certificate. Coming back to Dr. Saini, do you have any problem in unmuting yourself?
Dr. Junaid, you are getting my voice?
Yeah, now it's clear. Please go
Okay,
ahead.
okay, please. You can continue.
Okay, so the question, another question is, how does host microbiome influence growth of Leishmania?
Uh, in research experiment, it shows that microbiome of sandfly collected from the host means their means genome structure, and that sandfly which cultivate inside the lab, that both are two are controversial. So till now there is no any concluding remarks about how microbiome influence the establishment of infection inside the vector. There is no clear evidence. Means it's till now it's an controversial matter.
Thank you. The another question is asked by Devjani Chanda. What is the reason behind the indifferent distribution of Leishmania species, also causing atypical Leishmaniasis in one region and visual Leishmaniasis in another region?
Uh, that distribution of disease mainly caused due to the prevalence of vector species and circulation of the parasite. So where the circulation going on, there are those population are mainly found to get an infection. In Western Ghat belt, there are presence of sandfly population, and in other area, just like middle states of the country, there are sandfly population if somewhere are there, but parasite circulation are not there. So there those population are not getting infection, and they are not prone to getting infection. And in endemic zone, there are prevalence of vector population as well as circulation of the parasite are very high. They for that only they are more prone to get infection, and those area are denoted as an endemic zones.
Okay. There is another question. Why do human interaction with Leishmania not occur in Australia?
That the same answer means in that climatic condition, their vector population are not surviving. Means for that only in Indian that leishmaniasis is a tropical disease. Means in the tropical country mainly the vector are surviving and parasite circulation are there. But in other country means such type of Australia whatever you mentioned, that their climatic condition is not suitable for surviving the vector population, and that parasite circulation are also not there. For that only in one country they are endemic for this disease, and another country they are not getting endemicity.
There is a question asked by Arpita Datto. Could you please explain why and how do you select HSP as the marker for detecting the organism?
Sorry.
Could you please explain why and how do you select HSP as the marker for detecting the organism?
HSP have an conserved region. Means in these conserved region that can differentiate that species variation. For that particular region of HSP70 gene we selected, and we amplified for the diagnosis of these disease manifestations.
Another question in continuation with that, how can we relate the different HSP protein to the markers for different host and parasites?
In these aspects, several groups tried for such type of evidence, but only the HSP70, that particular region, are mostly suitable for that. For that only, people means researcher are working in this region, and other region means researcher tried, but it didn't give any sense for detection.
Now, probably the last question: how urea drugs like urea stibamine still relevant in Leishmania treatment?
Uh, that that drug also means have some adverse effect, but in these area, liposomal amphotericin B for the treatment of visceral leishmaniasis in a single dose 10 mg per kg body weight we are using for the treatment of visceral leishmaniasis. This is the first line drug of choice approved by WHO, and we are in government of Kerala, and we are treating those patients by single dose of this particular drug.
Thank you very much, Prasanta Saini. We have lots more questions, but due to time constraint, we have to complete this session here. We will send you the question so that you can get back to the participant. Thank you very much for being with us. We are ending the session here. We will be back by 11 a.m. with another interesting lecture by Professor Onivan Das. Thank you very much, all the participants.
Thank you so much, Junaid.
Welcome.